Hi, Carlos. Thank you so much for joining us. I think that this is going to be basically like a lightning round of all the big questions that everyone has about COVID, which I know we all have a lot. I guess just to start, I mean, we're now so many months into this pandemic. I feel like a lot of us thought that we would be done with it by now, but it looks like we might just still be starting. What do you know now about COVID-19 that you wish you had known 3 months ago?
Well, thank you, first of all, Olga, for having me, and it's a pleasure to be with you and all of the followers of your wonderful program. Look, this is a new disease. We didn't know anything about it, and we have learned quite a bit, but there is so much more that we need to learn. We were definitely not prepared. Um, we lack reagents. The chain supply was broken very early on. We were very frustrated because we saw a community in need. And as much as we were here, we were missing some of the basics for being in the kitchen. I mean, if you are going to cook and you don't have, all of the right ingredients, it's very frustrating. So we learned that we need to be better prepared in the context of making sure that some of the more garden variety reagents that we need in a lab need to be in supply, and they need to be ready in order to address problems when they come, rather than just running after them.
So something we've all heard a lot about in recent months is antibody tests, which are different, of course, than the actual coronavirus tests that you get to see if you're currently infected. Can you talk about what exactly antibodies are and what an antibody test looks like and what it shows?
Sure. So in general, people talk about diagnosis, and diagnosis in this case, it's identifying a pathogen, something that it's not usually on us, in this case, the sequence of a virus. So this is the diagnosis, and it's based on molecular genetics and identifying by PCR the viral strength. In here, what we are doing, it's rather not a diagnostic test, it's a monitoring test. It's a test that looks after you had the infection, if your body is building up an immune response. Nature has given us an enemy, but nature has given us a tool to combat this enemy, which are the antibodies that we produce. Now, the B cells take time. If you really have been infected and the B cells and your immune system has seen this new virus, they need time to produce these antibodies. And then they need time to produce a blood that is rich in these antibodies. So antibodies are our soldiers that go around looking for these enemies. They bind to them, and in the best of the situations, if they are functional, they can kill these pathogens. We call this process neutralization. So what our test does is that it looks for the presence and the quantity of these antibodies in the population in general, trying to do two things. One, to be qualitative, saying yes or no, you have seen the disease, so you have antibodies and we can now monitor you, or no, you don't have antibodies, you definitely didn't have the disease, as we can discuss it, and you have not seroconverted, meaning that if the disease is in front of you, you're going to be sick. You still don't have the armamentarium to attack that disease.
Can you have antibodies, COVID antibodies, without having had COVID or being sick with COVID?
The answer is yes and no, so let me be more specific. They are individuals, and it's a percentage that we are learning that we call asymptomatic individuals. They had the disease, they never really felt sick. Maybe a couple of days they felt that they were under the rain, but not sufficiently probably as to go to a doctor. They took it easy. And in these individuals, we have been now following some of them because they have come to us and the antibodies are present. So you can have antibodies even though you think that you have not had the disease. But in the cases where individuals have had the disease and they were positive for the viral tests, we have seen that essentially 99.5% of these individuals produce antibodies. And interestingly enough, some individuals that have been more sick, they produce more antibodies. And we now know that most of the times, these antibodies are functional antibodies, meaning that they can neutralize and they can kill the virus.
Huh. How long are you immune from the virus if you have antibodies?
Well, that's a question that we will need some time. What we can tell based on the experience of similar diseases in our history, that there are some responses, they are for life. There are some vaccines that you just receive it once in your life and you don't need to have it again because you produce essentially a very durable response. That's the case, for example, for measles or for polio. In the context of some other viruses that tend to mutate more easily, like the influenza, the typical flu, you need a reminder every year or 2 years. What we can say is that we were one of the first groups that tested based on both the diagnosis, identifying the virus, and the antibodies. And when we call back some of our first positive antibody responders to help us in donate plasma so they can donate this very rich antibody blood to patients that they are very sick. All of them after 3 to 4 months are still positive. The response seems to be at least for 3 or 4 months. We need time to keep following them. We have these studies under specific protocols, longitudinal studies, and our hope is that they will be here most of the, at least in most of the situations, if possible, like the influenza, which is at least 9 months to a year and a half.
So I have been wanting to visit my family. I'm sure other people, as this kind of social distancing wears on, they're going to want to see their family in faraway states and places. What's the safest way to do that? Is it to get a COVID test before you see them? Is it to get an antibody test? Is it just assume you have it and quarantine for 2 weeks beforehand? How should we be seeing, like, if it's safe for us to be around other people?
Well, one of the problems that we have had during this period, it has been testing. Everybody wanted it to be tested, and we were lacking, again, from reagents to equipment to test all of the people that wanted it to be tested. Nowadays, with the ease of the virology, the detection of the viruses test that can be done in a matter of minutes, if not a little bit longer, testing for the presence of virus, I think it's important because you want to know if you are infected or not, because again, you may be asymptomatic. You may not even feel that you are sick, but you may, and then you may be exposing other individuals that they are more fragile or individuals at high risk, immunocompromised, or the elderly that have other comorbidities such as diabetes or some other cardiovascular conditions to be exposed. So testing for the virus, I think it's important if one wants to go and visit a community. Testing for the antibody is going to be critical for many reasons, because we want to know if you have seen the disease. We want to know if you need to be vaccinated immediately if a vaccine comes available, because if you already have antibodies, you are protected. And more importantly, one of the things that we believe we have done, and it's part of our contribution, is that we didn't just say yes or no. I mean, there are two kinds of assays, the ones that we call qualitative, yes or no, and the ones that can be quantitative in metrics. So we have learned that the more virus you have, what we call viral load, the more sick you may get. And also the more antibodies you have, the more protected probably you are. And definitely you are a better donor for these kind of protocols for convalescent plasma, that they are definitely helping individuals that are still very sick.
We are running out of time, but I have a burning question, which I'm sure other people have, which is, when do you think we will have a vaccine that everyone can get?
Well, there are several clinical trials that they are ongoing, and some of them are already moving from phase 2 to phase 3, and even one in late phase 3. Results are very encouraging. This needs now to be tested at a different level. So we believe that hopefully by the beginning of 2021, we may have vaccines that could be at least validated and start being produced for mass use. And maybe one of the things that I just want to add is that this is a disease of two diseases. It's a disease that it starts with the respiratory system, that affects the respiratory system. and you get the typical flu-like. But this virus, it's a very peculiar virus. We have been able to do a lot of post-mortem analysis, autopsies, and we are learning that after a certain period of time, some of these viruses have learned how to also attack the endothelial cells and the cardiovascular system, moving from a very specific kind of disease to another disease that needs other treatments and other modalities of therapeutics. So important to have that in mind, because this is part of the complexity of this COVID-19 disease that, again, we have learned quite a bit, but there is so much that still we need to learn.
Absolutely. Well, thank you so much, Carlos. I really appreciate your time today. I'm sure we all do. And I will toss it back to the showrunners here.
Thank you very much, Olga. Thank you.